So this is chapter 2, but it's not the microbial world in you. This is chemistry. So this right here is the Bacillus anthracis bacteria, and so we more commonly understand this to be anthrax. And so what this bacteria will do is it will create what we call a spore. And these spores are not readily digested. But how does it do that? And there's definitely some chemical things that occur there to make it a hardened kind of cell that can lie dormant for a very long time. You know, if you were around after 9/11, they were worried about people sending anthrax in the mail, and then it's happened a couple of other times since then. And so when they say, oh, you know, there's an anthrax scare, they're talking about the spores being sent out. And so this is an example of that. And, you know, the chemistry of how it actually creates that is actually pretty interesting. So let's take a look at the structure of atoms. So this is our learning objective. We're going to describe the structure of an atom and its relation to its physical properties of the element. So chemistry is the study of interactions between atoms and molecules, and the atom is the smallest unit of matter that cannot be subdivided into smaller substances. Atoms interact to form molecules. Atoms are composed of 3 different things: electrons, protons, and neutrons. Electrons are negatively charged particles, protons are positively charged particles, and neutrons are uncharged particles. And we'll probably go over this later, but I'd like to mention right now, before we go on, that these essentially have no mass. The electrons are not going to have any mass. These are going to have a mass of 1 AMU, the protons there, and the neutrons are also going to have a mass of 1 AMU. And so this will come into play a little bit later when we talk about isotopes. So protons and neutrons, these are going to make up the nucleus, and the electrons are going to move around the nucleus. And so here we can see this diagram. This is a Bohr model of the atom, so BOHR. If you've had chemistry, and I'm sure you've had chemistry in at least one of your classes, like at least a chapter on it, you'll know that there's different models and ways to look at this. And so once you start to understand a little bit more, you understand that this is a really easy way for us humans to see this, but it's a terrible way that it actually occurs. And so we've got these electron shells orbiting our nucleus here. And so we've got protons and we've got neutrons in the nucleus. So here's our proton right there. And then our neutron we're going to color here in a different color. And so the different numbers of protons and neutrons, the different number of protons are going to determine what element it is. And then the different number of neutrons are going to determine the isotope of that. So chemical elements, the atoms with the same number of protons are classified as the same chemical element, and each different chemical element has a different number of protons. And so this is the atomic number, the number of protons in the nucleus. I like to think about this as the identity number. And so the atomic weight is the total number of protons and neutrons in an atom. And this is where we get isotopes. So here are 3 different isotopes of oxygen. You have oxygen 16, oxygen 17, and oxygen 18. They all have 8 protons, but they all have different numbers of neutrons. And so you can figure out what type of isotope you have by subtracting the mass number, which is here on the top, from the number of protons here on the bottom. So if you realize this, so the nucleus is the protons plus the neutrons, and this is equal to the atomic mass. If you rearrange this this formula, you get that atomic mass, or AM, minus the protons is going to give you the number of neutrons. Okay, and that's actually a pretty easy formula to do, so don't be scared to do it. Now, these different isotopes are going to have different properties. A lot of isotopes are radioactive. These aren't the best isotopes, and also I'll note that this is not a common notation. This is a very old notation. A lot of times you're going to see it in different notations, and so just be aware that that is kind of an old and outdated notation there. All right, so here are the elements of life. We've got hydrogen, carbon, nitrogen, oxygen, sodium, magnesium, and phosphorus. I like to think of the very important ones that we have in our bodies, SCHON. Let me erase that. That was a terrible N. NPS. Okay, so we've got sulfur, we've got carbon, we've got hydrogen, we've got oxygen, we've got nitrogen, and we've got phosphorus. And so that's how I remember these important ones. We are mostly carbon. Okay, we've got a lot of carbon in there. We've also got a lot of hydrogen and a lot of oxygen because we're made up of a lot of water as well. Okay, but we're going to find slight differences between the microbial world and us. Okay, so continuing on, here's some other elements of life: sulfur, chlorine, potassium, calcium, iron, and iodine. So these are very important. Hydrogen, carbon, nitrogen, and oxygen are the most abundant chemicals in living organisms, and in pretty much all living organisms. So electrons are arranged in what we call electron shells. These correspond to different energy levels. And so here's a check your understanding question, and it's asking you how carbon-14 differs from carbon-12. And so what is the atomic number of each atom, and what is the atomic weight? And so the atomic number here is 6. So atomic number is 6, and the atomic weight of each of them this is going to be 14 and 12 respectively. Okay. Okay, so when they say carbon-14, this is what they're talking about. This is what they use for carbon dating. So next we're going to talk about bonding and how that kind of gets us to making molecules. So atoms form molecules by combining to fill their outermost shells. And the numbers of missing or extra electrons in the outermost shell is known as the valence. And so molecules hold together because the valence electrons of the combining atoms form attractive forces called chemical bonds between these atomic nuclei. And so a compound is made of a molecule, compound is a molecule that contains 2 or more types of atoms. So water is 2 hydrogen atoms and 1 oxygen atom, and it comes together and it forms a compound. Okay. The ionic bonds, these are the number of protons and electrons are equal in an atom in general. Ions are charged because they've either gained or lost an electron. And so here we'll talk about how we form ionic bonds. All right, so sodium has 1 electron in its outer shell. You can see it right here. Okay, and so that electron in the outer shell is going to be gained by the chloride ion, which is going to have 7 electrons in its outer shell. They want to get to 8, and this is known as the octet rule. And because they want to get to 8, and then they're going to be electromagnetically attracted to each other. And this is why we get table salt. So cations are atoms that lose electrons and become positively charged ions, and anions are negatively charged ions. How do I remember this? Well, someone, when they made this up, decided to put a plus sign here in the middle of the cation, and cations are positive. So that's how I remember that a cation is positive and the anion is negative. So ionic bonds are attractions between ions of opposite charge. One atom loses the electron and another gains an electron. And so we just saw this, and so this is that bond, and so we create these 2 different charges here, and then they stick together through the electromagnetic force. Covalent bonds form when 2 atoms or more share pairs of electrons. And so these are going to be ones that are going to share it, and they're either going to share it equally or they're going to share it unequally. And so covalent bonds are stronger and more common in organisms than ionic bonds. So here, hydrogen in its natural state exists as a diatomic molecule because it doesn't have an outer shell that's completely full. And the first outer shell, so I remember how I told you a couple of slides ago that the outer shell had 8 in its outer shell? Well, the very first shell only has 2 electrons in its outer shell. And so these are going to come together and they're going to form this covalent bond. Other covalent bond formations might happen between carbon and hydrogen. This is how we get methane. And as you can see, they are sharing electrons in these places. And so each of the hydrogen ions is going to bring one of these electrons, and then and then the carbon atom is going to bring the other electrons, and they're all going to get together and share those electrons so that they all have a full outer shell. So hydrogen bonds form when a hydrogen atom is covalently bonded to an oxygen or nitrogen atom, is attracted to another nitrogen or oxygen atom in another molecule. Hydrogen bonds are very useful because they are very weak bonds. They're strong enough to hold something together, like your DNA, but they're also weak enough to where they can easily be split. Okay, so you guys have probably put a water bottle in the freezer before. And when you put that water bottle in the freezer, you find out after it goes completely frozen that it bulged, it got bigger. And one of the reasons that happens is, well, we have these hydrogen bonds between our oxygen atom here and the other hydrogen atoms because they have a slight negative charge. This is because of a polar covalent bond where the electron is going to spend more time in different places. And so they're going to have to make this lattice structure, and this is one reason why ice floats on water. And so it's a very important concept for biology that hydrogen bonds are associated with water as well as some other things. So molecular weight in moles. The sum of the atomic weights in a molecule is the molecular weight. And 1 mole of a substance is its molecular weight in grams. Okay, so we take H2O, we find out that the oxygen has a molecular weight of 16, and the hydrogen has a molecular weight of 2, because there's 2 molecules in there. And so 1 mole of water weighs 18 grams. So here's a check your understanding question. Make sure you guys take a moment to take a look at this. So let's talk about chemical reactions. And so we're going to diagram the 3 basic types of chemical reactions we're going to see. So a chemical reaction involves the making or breaking of bonds between atoms. A change in chemical energy occurs during a chemical reaction. An endergonic reaction absorbs energy, and an exergonic reaction releases energy. So synthesis reactions occur when atoms, ions, or molecules combine to form new, larger molecules. So we're taking our reactants here, we're taking A, we're adding it to B, and they're going to combine in some form to form a new product, AB. And so this is what we call anabolism. This is the synthesis of a new molecule in the cell. And generally, you should be understanding that this is going to take energy. And so this is what we consider endergonic. Okay, so this is going to require energy. In a decomposition reaction, the opposite is occurring. We're taking these huge molecules, or these bigger molecules, and we're breaking them down into their constituent parts, A and B. And this is catabolism. So this is the decomposition reactions that are going on in a cell. Generally, this is exergonic. And so it gives off energy. The last one we'll talk about is an exchange reaction. And so these are part synthesis and part decomposition. And so we have, in this case, we have sodium hydroxide mixing with hydrochloric acid. Okay, so this is the classical strong base, and this is the classical strong acid. And so what happens is you put these two things together, you start to get sodium and chloride, which we talked about a little bit earlier, that's table salt, and then we get water. Anytime you mix an acid and a base, you always get a salt and water. So chemical reactions are also reversible. And so most reactions, especially in biological systems, are reversible. And they can readily go in either direction. But they might need special conditions. So if we have reactants A and B here, and we add heat to it, we might get the product AB. If we add water to this, it will go back to having A plus B. And so these can go back and forth, and as long as we have everything we need, we will have these reactions occurring until they reach equilibrium. So here's your check your understanding question. This is a chemical reaction below is used to remove chlorine from water. What type of reaction is it? So here's our learning objectives. We're going to list several properties of water that are important for living systems, and we're going to define acid, base, salt, and pH. So water is inorganic. It's a polar molecule, meaning it has an unequal distribution of the charges of the electrons and protons in it. It's also a great solvent. The polar substances undergo dissociation in water. They form solutes, and hydrogen bonds absorb a lot of heat. And so here we can see the hydrogen bond formation. And so you can see here how we have a few dipoles. We have the positive ones by the hydrogen ions, and we have the negative ones by the oxygen atoms. So we have already taken a look at the hydrogen bond formation in water, and this is why your ice floats on your water. So water acts as a solvent for sodium chloride as well as lots of other things. And the reason why is when you drop sodium chloride into water, it starts to dissociate and form what we call hydration layers. And so the chloride ion gets dissolved in the water because you have the positive charged hydrogen being attracted to the negative charge of the chloride ion. And the same thing goes for the oxygen atoms and the sodium atoms there. And so they're going to be buffered and they're going to dissolve. So when you put a little bit of salt into water, it dissolves. Okay, and so this is what's happening there. So substances that dissociate into one or more hydrogen ions and one or more negative ions are going to be what we call acids. Okay? And so hydrochloric acid, when you drop it in water, is going to fully dissociate because it's a strong acid. And strong acids are different than weak acids. Weak acids are only going to partially dissociate, and strong acids are going to be fairly simple. But we're not going to go too far into that because I don't think we're going to need that much depth on it. And when we do, we will definitely cover it. But anytime you see a hydrogen ion, you should be thinking that this is an acid. Okay, so you put hydrochloric acid into water, and it's going to dissociate, and it's going to make the water more acidic. Substances that dissociate into one or more hydroxide ions are what we call basic. Okay, another word for this is alkaline. And so sodium hydroxide is going to be dropped into water, and you're going to get sodium and the hydroxide ion splitting in the water. So here's just a picture of that. And so substances that dissociate into cations or anions, neither of which is hydrogen or the hydroxide ion, the OH minus there, are what we call salts. And so sodium chloride is a salt. So you guys have seen how it forms hydration layers. This is exactly what's going to happen here. Okay, so the concept of pH. This is an interesting concept, and it does involve a little bit of math, but it's an important bit of math because once you understand it, you will completely change how you feel about pH. Okay, so the concentration of hydrogen ions is in a solution is going to be expressed as pH, and pH is equal to the negative log base 10 of the hydrogen ion concentration. So when you increase the hydrogen ion concentration, you increase the acidity or you decrease the pH. Okay? The increasing acidity, or the OH minus, increases that alkalinity. And so when you have an increase in alkalinity or a decrease in acidity, you have an increase in pH. And so most organisms grow best between pH 6.5 and pH 8.5. All right, so the pH scale is here. You can see the acidic solutions there at the top. So stomach acid is a little bit over 1. Lemon juice is right at 2. Grapefruit juice is 3. Wine or tomato juice is about pH 4. Urine is around a pH of 6. Generally, it's in a decent range depending on what you're eating and how much water you're drinking. Milk is about 6.5. Pure water is right at 7. And so at 7.0, the amount of the concentration of hydrogen ions is equal to that of the hydroxide ions. And so it's completely balanced there. Human blood generally rests between 7.35 and 7.45. Salt water is about 8.5 as a pH. And as you continue to go up, you get through ammonia, and then household bleach, and then oven cleaner, and then they say lime water. I'll point out that the classic strong base is sodium hydroxide, and then the classic strong acid is going to be hydrochloric acid. And so in their pure forms, these are at pH 0 and pH of 14. So here's your check your understanding question. Make sure you guys understand what's going on here. So let's talk about organic compounds. And we're going to distinguish organic compounds from inorganic compounds, and then we're going to define the functional group. So what is an organic compound? An organic compound commonly contains hydrogen, oxygen, and or nitrogen in addition to carbon. It must have carbon to be considered organic in regards to chemistry. So the chain of carbon atoms in an organic molecule is what we call the carbon skeleton. And so functional groups bond to the carbon skeletons and are responsible for most of the chemical properties of a particular organic compound. So here we see methanol, we see ethanol, and we see isopropanol. If you have methanol, you shouldn't drink methanol. Okay, don't drink. Okay, and the reason why is it will make you go blind. Okay, but the functional group here is the alcohol, or the hydroxide ion. Okay, ethanol, this is the one we can drink. So make sure you drink in moderation, only if you are of age. And so you can see the functional group here is the alcohol. In isopropanol, you see the functional group here. You probably don't want to drink this one either. I don't think it's going to taste very good. So there's some other functional groups here. We see the aldehyde group, we see the ketone group, we see the methyl group, and we see the amino group. And so these are the ones that we're really concerned with in biology. And there's a couple of more that are important. Okay. Continuing on, we have esters, we have ethers, we have sulfhydryls, we have carboxyl groups, we have phosphate groups. So you might hear carboxylic acid. That's where you're going to get that guy from. Phosphoric acid, that's where that one's going to come from. Sulfuric acid is not going to be like this, but it's going to have some sulfides in there, and it could easily create sulfhydryl kind of functional groups. Ethers, these are very flammable, so be very careful with those. Esters smell very good. A lot of the things that you will buy at Bath and Body Works or anything that smells pleasant is generally giving off some type of ester. So identify the functional groups in an amino acid. So I will show you here that this is the carboxylic acid. So this is the carboxyl group. Okay, and so that one is going to be in more simple chemical terms, COOH. Anytime you see that, you should be thinking that's the carboxyl group. Here is the amino group. Okay, and that's generally notated in H2. Then we have the R group, and the R group is changeable, and we'll see this when we talk about the amino acids and how they work. So small organic molecules can combine into large macromolecules, and macromolecules are polymers that consist of many small repeating molecules called monomers. And monomers join by dehydration synthesis or condensation reactions. And so what we get here is we get the hydroxide ion here and the hydroxide ion here, and what we get is the R and the R prime coming in here, and they bond, and then we get the H2O right there. And that's how we get that. This is what we call dehydration synthesis. We are giving off water. Okay, and so here's your check your understanding question. So make sure you guys take a moment to take a look at these. So let's talk more about organic compounds. And so we're going to do a lot of identification, and then we're going to describe some very important ones that are important for biology. So carbohydrates serve as cell structures and cellular energy sources. These include sugars, starches, and consist of carbon, hydrogen, and oxygen with the formula CH2ON. And so many carbohydrates are what we call isomers, molecules with the same chemical formula, but they have different structures. And so monosaccharides, these are very simple sugars with 3 to 7 carbon atoms. Glucose and deoxyribose are examples of common monosaccharides. Disaccharides are formed when 2 monosaccharides are joined through a dehydration synthesis. And so disaccharides can be broken down by hydrolysis. And so here's an example of hydrolysis. So what you do is you take that glucose and fructose, so what this is sucrose. Okay, and you might put that in your coffee in the morning, or you might heat that up in a pan with some water to make some sweet syrup for your bar. And so the dehydration synthesis is going to bring these together and give off the water. Okay, so the dehydration synthesis is going that way. And so I'll do it in a different color so you guys can see it going the other way. The hydrolysis reaction is when you add water into it and it splits off. And so you're adding the H2O in here so that you can get those things to split. So polysaccharides consist of tens or hundreds of monosaccharides joined through a dehydration synthesis. Starch, glycogen, dextran, and cellulose are polymers of glucose that differ in their bonding and function. So you might be familiar with starches. Starches are very tasty if you fry them up, especially if they're potatoes. Glycogen. Glycogen is the storage form of glucose in the human body. So we store this in our muscles. We store a lot in our liver. And then cellulose, you guys have probably had some salad sometime, and everybody says, "Oh, salad's so good for you because it has so much, fiber." And this is where that cellulose comes in. Our bodies can't naturally break this down, but we have bacteria in our guts that can break this down. So lastly, dextran is probably the weirdest one out of all of these. Dextran is a sugar that's found in wine, and it's also found heavily in dental plaques. So that plaque that's made on your teeth is very thick, and one of the reasons why is that's one of its chemical properties, and it's very hard for when they're producing sucrose. Sugar and refining it, getting the dextran out is actually a very difficult process. So here's your check your understanding question. So make sure you guys take a moment to take a look at that. So what are lipids? Lipids are the primary components of cell membranes. They consist of carbon, hydrogen, and oxygen. They are nonpolar and insoluble in water. And so simple lipids, these are fats or triglycerides, and they contain a glycerol and fatty acids. These are formed by dehydration synthesis. And so here we've got our glycerol right here, and then we've got our fatty acid right here. And so this is the structural formula of a simple lipid. A saturated fat has no double bonds in the fatty acids, and an unsaturated fat is going to have one or more double bonds in the fatty acids. And this is where we get cis and trans. And so what's going to happen is if it's cis, that means it's going to look like that. Okay, they're going to be going in the same direction. But a trans double bond is going to be going in different directions. Okay, so they have opposite sides of the double bonds. And so these are going to cause different chemical properties. And so here we can see we have a cis configuration where the hydrogen atoms right here are in the same on the same side. So complex lipid contains carbons, hydrogen, and oxygen, as well as phosphate, the nitrogen, and or sulfur. And so cell membranes are made of complex lipids called phospholipids. So glycerol with two fatty acids and a phosphate group. And these phospholipids have polar as well as nonpolar regions. So the polar region is going to be up here. Okay, so this is the polar head, P-O-L-A-R. And so then we have the nonpolar tails. And generally when we have a phospholipid bilayer, we see that the fatty acids, the non-polar sides, are going to be facing each other. Okay, so we have different structures based on what kind of things are happening with the things. And so saturated fatty acids are going to be very closely packed. Unsaturated fatty acids are going to be very loosely packed, as you can see here. And then sterol molecules separate the fatty acid chains, and this helps to maintain rigidity in the cell membranes. Steroids are 4 carbon rings with a hydroxyl group attached to at least one ring, and the part of the membranes that keep the membranes fluid. Okay, so here we have the cholesterol rings. So we have A, B, C, and D. These are the rings, and so this is cholesterol, a steroid, and this is one that we find in our cells in the cell membrane. Now, this isn't the cholesterol that you go get checked when you go to the doctor. They're looking for low-density lipoproteins and high-density lipoproteins. So here's a check your understanding question from Module 2.9. So proteins are made of carbon, hydrogen, and oxygen, as well as nitrogen and sometimes sulfur. And they're essential in cell structure and functions, and enzymes that speed chemical reactions. They're transporter proteins that move chemicals across membranes, and flagella that aid in movement. Some bacterial toxins and cell structures are also made of proteins, and so all of these things are very important for normal functioning of a cell. And so proteins consist of subunits called amino acids, and these amino acids contain an alpha carbon that has attached a carboxyl group, the amino group, and a side group. So here you can take a look at the structure of an amino acid. You can see the alpha carbon there in the center, and attached to that alpha carbon are four different groups, or essentially atoms. And so the side group is attached there, and the cyclic side group here makes it a tyrosine. You also have the carboxyl group, which we talked about a little bit earlier, as well as the amino group. And so here are the 20 amino acids that are found in proteins. And so these are the amino acid names, including the 3-letter abbreviation, their structural formulas, and their characteristic R group that is shaded in green. Note that cysteine and methionine are the only amino acids that contain sulfur. So make sure that you guys take a look at these and appreciate these. I'm not going to expect you to necessarily memorize these, but it's one of those things that you should at least be aware of them. One of the interesting ones that I will point out to you is proline. Okay, so proline has this cyclic shape, and proline is, when it's produced, it doesn't cyclize until after it gets produced and then dropped in a certain condition. And so that's going to be one of those things that is kind of important. So this is the only one that cyclizes on itself, and it looks different than all the other ones. And so it also disrupts a lot of protein folding, and so that's why it's a very important amino acid. So they either exist in 2 different stereoisomers, and so we probably haven't talked about this much in the past. The L forms are the most often found in nature. The D forms are not really that useful to our body for a lot of reasons. And so what L and D are kind of they're the levorotary and dextrorotary, and they came up with these words when they figured out that there were different isomers for amino acids and several other and many other chemical compounds. And so it's kind of like your right hand and your left hand. They look very similar to each other. In fact, they're mirror image, but they're in a different configuration. You can't fit your right hand into a left-handed glove unless it's upside down. So it's one of those things where you just can't do these things because there is a different configuration. Peptide bonds are found between amino acids are formed by dehydration synthesis. This is an example of a covalent bond. And so here we see this dehydration synthesis, so we have a glycine and an alanine coming together, and you'll see the carboxylic acid group and the amino group are coming together, and then they're going to kick off this water, and we're going to get our peptide bond between those two. So the primary structure is the sequence of amino acids in the polypeptide chain. So I'm going to write the sequence of amino acids. Okay, and this is indicative of the primary structure that you're going to see. And so here we can see that primary structure. We have peptide bonds, and on one end we have the carboxylic acid, and on the other end we have the amino group. The secondary structure wins when the amino acid chain starts to fold and coil into a helix or a pleated sheet. And so this is only because of the chemical properties that we see there. And so the pleated sheet, they might call them a beta pleated sheet in some literature. And so they start to kind of fold on themselves, like if you've ever seen an accordion, it's got the bellows and stuff like that, and it's covered in this thing that can stretch and then compress and stuff like that. The alpha helix, this is going to create some spirals, and so when you look at a protein, you might see a spiral or some type of sheet like that, and that's what they're talking about. Tertiary structure is when the helix or sheet folds irregularly, forming disulfide bridges, hydrogen bonds, and ionic bonds between amino acids in the chain. And so this is when the protein becomes functional. And if it doesn't fold correctly, it's not going to be functional. Okay, it has to fold correctly, and it has to have all the other prosthetic groups put in for it to take that tertiary structure. The quaternary structure consists of two or more polypeptides that have come together. And so here we can see the relationship between these two. You'll probably hear this talked about a lot in biology when they're talking about hemoglobin. And so hemoglobin is a protein that has 4 different subunits, and they come together and they create that quaternary structure. And so here are the levels of protein structure. We start with primary structure, which is the sequence of amino acids. The secondary structure is those beta pleated sheets or the alpha helixes. And then the tertiary structure is when it folds into some 3-dimensional shape. That's when it becomes useful. And then lastly, we have the quaternary structure, was when it gets together with other proteins. So proteins can undergo what we call denaturation. And denaturation occurs when a protein encounters a hostile environment, such as a high temperature or a low temperature, as well as high or low pH. And therefore, it loses its shape and its function. So one of the best examples of this is, cooking an egg. So you guys have probably cracked an egg before and put it into something, maybe you put it into a cake batter or something like that. Or if you cooked an egg before and you put it in a pan and you fried it up, you'd probably notice that it's different than it came out of the egg in. And so for this reason, you're never going to get those proteins back into their original structure. There's just no way to do that. So conjugated proteins consist of amino acids and other organic molecules like glycoproteins, nucleoproteins, and lipoproteins. So here's your check your understanding question. Please make sure you guys take a moment to take a look at this. The last thing we're going to talk about, here is nucleic acids, and these consist of nucleotides. And nucleotides consist of a 5-carbon or a pentose sugar, and a phosphate group, as well as a nitrogen-containing purine or pyrimidine base. And so nucleosides consist of a pentose and a nitrogen-containing base. So DNA is deoxyribonucleic acid. It contains deoxyribose. It exists as a double helix. The adenine hydrogen bonds with thymine, and cytosine is always going to hydrogen bond with guanine. And the order of the nitrogen-containing bases forms the genetic instructions for the organism. So here you can see this, and so there's a lot to really unpack here, and so just take a moment to appreciate this this particular drawing. So you can see that it's on a phosphate backbone. So you can see they've labeled the sugars here, but it comes down here and it coils. It's a helix. Okay. You'll also notice that between C and G they've got 3 hydrogen bonds, and between A and T they've got 2 hydrogen bonds, and they're showing you up here in a chemical formula how that is actually occurring. And so these carbon atoms are important. These are one of the largest molecules that we have in our bodies, and so they're very important for storing that genetic information. The ribonucleic acids contain ribose, they're single-stranded, and adenine is going to bond with the uracil. Okay, so it's different. And cytosine is going to bond with guanine still. And so there's several different types of RNA that play a specific role in protein synthesis, and we will talk about that when we get to that chapter. And so here you can see that you've got the ribose sugar instead of the deoxyribose sugar, and then you've got uracil, which is unique to RNA. So here's your check your understanding question. So make sure you guys take a moment to answer that. Adenosine triphosphate is made of a ribose, adenine, and 3 phosphate groups. And so here you'll see the structure. Okay, so you've got the adenine is right here, you've got the ribose sugar, which is right here, and then you've got your 3 phosphate groups over here. What I'll tell you is this first bond holds a ton of energy. Okay, and so that's why it's a very useful energy storage molecule. The second bond doesn't hold as much energy, but it still holds some, so it still can be taken off there. Another interesting fact that I'll put in there for you is magnesium is used to stabilize this molecule because it's a very unstable molecule as it is. It wants to get rid of it, okay? Most chemicals don't like to be in their high energy state for very long. So adenosine triphosphate, it's going to store that chemical energy that's released by some of the chemical reactions, and it's going to release phosphate groups by hydrolysis to liberate useful energy for the cell. So usually the way I write this is ATP plus H2O is going to give us ADP and the inorganic phosphate. Okay. Now this isn't a perfect chemical formula. I understand that there's a lot more that goes into this, but what we want to see is we want to see the adenosine diphosphate here, giving off that inorganic phosphate right here. Okay, and so that can happen in a lot of different ways. So here's your check your understanding question. Make sure you guys take a moment to look at this. Make sure you guys review this. If you have any questions, please get in touch with me.